Good morning, students. Today we are going to discuss about structural elucidation techniques. So in our syllabus, we have three different techniques. One is mass spectrometry, then X-ray diffraction. Third one is nuclear magnetic resonance. So here today we'll be going to discuss about mass spectrometry in detail. So what is mass spectrometry? It is an analytical technique which is used to measure mass to charge ratio of the particle. Okay. So this is the technique which will be help us to identify the unknown structure of a compound or studying the molecular structure of a compound. So here it will be used to determine the mass of the particle and also determining the elemental composition of the molecule or a sample. So the mass spectrometry principle will be consist of different sectors. One is ionization of a chemical compound. Because of this ionization, different molecular fragments will be produced or charged molecules will be produced. According to the production, we can detect the mass to charge ratio of the compound or a molecule. So you can see this structure. So here you can able to see propane is there, CH3, CH2, CH3. So if you are ionization this compound, then you can able to get different fragments. Now you can see in this next diagram. So here fragment, three different fragments of different molecular weights you can able to see. First one is CH3, CH, CH3 plus. In second one is CH3, CH plus. Third one is CH3 plus. So these are all the compounds that can be produced after the ionization. Okay, here the what is the basic thing will be the substance will be hit by the different electron beams. Okay, then it will produce molecular ions. While we are applying magnetic field, these molecular ions are accelerated and it will reach the detector at different time intervals by different curved path. Okay, so in this diagram, you can able to see Fm is equal to Fc. What does it? The magnetic force is equal to the centripetal force. So here you can rearrange the equation and finally you can able to get R. R is relative intensity. R is equal to M into V by Q into B. This M by Q is the mass to charge ratio. So this R directly proportional to M by Q. So here you can able to see in this down the diagram, there will be the graph or a peak will be there. So here it will result relative abundance versus M by Q. So M by Q is directly help us to find out the molecular weight of the compound. You can able to see 15, 28, 43, 44. So these are all the different ions that molecular weight will be there. So here you can able to find out the unknown compound. Okay, so here it will measures relative abundance and which is going to find out the M by Z ratio. So because of this M by Z ratio, we can find out the molecular weight of the ion. So we can detect unknown compound, which compound will be there. Okay, so here you can able to see. So here two different ions it can be produced. One is molecular ion and other fragment ions. So which will be having higher molecular weight in the final profile. So which will be called as molecular ion peak or a highest peak value. Okay, so other peaks, other fragments are data fragments we can able to call, that is 15, 28 and 43. These are all the data peaks we can able to get in the chromatogram of the mass spectrometer. So this is the basic diagram of the mass spectrometer, basic instrumentation you can call it. So here three, four different sections will be there. First one is ionization, second one is acceleration, third one is detection, reflection, and final one is detection. So here the vaporized samples will be coming here. So the vaporized vapors will be there. Okay, the sample in the form of vapor, it will be going into the ionization chamber. There it will get produced, it will be converted into a ions. So these production of these ions will be happening in the ionization chamber. Then it will go to the acceleration chamber where all the ions will be accelerated and it will be passing in the same energy. Okay, same speed it will be going. and third sector will be deflection. So here only we'll be applying strong magnetic field. So because of this magnetic field, according to the mass by charge ratio of the particle, so different time intervals it will be reaching. You can able to see different curved path will be there. Okay, so this will be happening in the deflection part. Then final will be detection. So these different time interval according to the mass to charge ratio, the ions will be reaching the detector and the profile will be making. Okay, so the amplifier is there. Will amplify the signal, and we can next we can reach the, so then we can analyze the which compound it is. So now I'll be showing one video. You can go through this video. You can understand it in clear. So you can see here, ions will be, I mean vapors will be there, and it will be going in the ionization chamber where ions will get produced. So all the ions will be going to the acceleration plates. It will be reaching the third part, deflection chamber. So here magnetic field will be applied and base to charge ratio will be reaching the detector. So different color will be there now, that green color, purple color and yellow color. So this will be different ions. So based on the mass to charge ratio, different time intervals it will be reaching the detector. Finally, you can able to see the mass spectrogram. Okay, so according to the mass to charge ratio, different profile you can able to get, and we can analyze the which compound it is. And if you go to the main instrumentation part, so there will be four components in the MS. First one will be ionization, second one will be acceleration, third deflection, and final will be detection. Okay, so here the third part, deflection will be there, no? So here three different compounds will be used. One is magnetic sector analyzer, second one is time of flight analyzer, and third will be quadrupole analyzer. So these are all the different types of analyzer which will be used to find out the mass to charge ratio of the ions which will be coming inside after the acceleration chamber. Okay, so here after injection of the vapors or a sample, so the vapors will be converted into a ionized particles. So that will be happening in the ionization chamber. Next will be acceleration. So what are all the ions will be produced? So all the ions will be accelerated and they'll be having same energy. So this is process will be happening in the acceleration chamber. Third will be deflection. So here, according to the mass to charge ratio, so the all the accelerated ions will be reaching to the detector. Okay, so here the deflector three different types of deflector will be there. So one by one we'll see later. And last will be detection. So what are the ions which will be finally strike on the plate that will produce generate or that will produce or generate a current. So that will be detected with the help of a detector. So this is one more diagram which will which will show the main instrumentation part of the mass spectrometry. You can able to see here all the things we have discussed before. No, everything will be there in this diagram. So you can able to see ionization area. So where ions get produced and the accelerating two different plates will be there. No, so this is the acceleration chamber, and next this will be the deflection part. So where strong electromagnetic plates will be, electromagnetic field will be applied here. So here you can able to see different curved path will be there. So this will be based on the mass of the compound. So if the mass of the ion or ion will be small, it will be initially the curved path will be very less. Then according to this charge of the mass, the different curved path will be there. Then finally it will be reaching the collector and the detector will be there. It will analyze and produce a signal. Okay. Then the major principle will be there. So here the amount of sample will be used in mass spectrometry will be very, very less. So small amount of a compound or a sample which we are going to analyze, it will get vaporized first, and the vaporized sample will be sent to the analyzer, sorry, ionization chamber, where it will produce small ions with the help of strong electron beam that will be there inside in the form of heated cathode filament. Okay, it will be there inside. So because of this, ions will be produced. Okay, then These ions mostly the ionization is achieved by inductive effects. Okay, so we can able to get mostly positive ions. So these positive ions are forced into out of the ionization chamber, and then they'll go to the acceleration chamber. So where with the help of electrostatic field they'll be accelerated and they'll get same energy. Okay, then the separation ions will be take place in the analyzation chamber. So we are applying strong magnetic field over here. So the pressure of this system will be 10 per minus 8 millibar. So because of this strong magnetic field, it will be applied perpendicular to the motional direction of the ions. So because of this, due to Lorentz acceleration, so according to the mass to charge ratio, different curved path will be there. So then we can able to differentiate different ions. Okay, so the ions with a different mass to charge ratio are forced to the exit slit. So where the variation will be there, and because of this magnetic field as well as the charge to mass to charge ratio of the ions, we can able to get it can able to reach the detector at different time intervals. So once it reach the detector, the current will be produced, and we can able to get different chromatogram or a profile we can able to get. Okay, after the ions will be passed to the exit slit, then they collide in the collector electrode. So because of this, resulting current will be there. That will be amplified and it will be registered. That means it will be recorded as the function of magnetic force, or and you can tell accelerating voltage. So here the first step will be ionization or a fragmentation, you can tell. So this will be taken place with the type of two different techniques. One is hard ionization techniques and next one is soft ionization techniques. So in hard ionization techniques will be their handling energy will be higher, so you can able to get more fragments, but the molecular ions will be lesser. So fragmentation will be higher, so molecular ions will be produced in lesser quantity. In case of second technique, soft ionization technique, the energy will be lowest energy will be handling, so the fragmentation will be lesser, so you can able to get more molecular ions. So already we have discussed now what is the molecular ion, what is the daughter ion. So in hard ionization technique, you can able to get higher daughter ions, lesser molecular ions. But in second case, the vice versa. So the methods will be there to produce ions. The gas phase method will be there and desorption method will be there. Then evaporative method will be there. So in case of gas phase, two different ionization will be there. One is chemical ionization, another one is electron ionization. In desorption techniques, the field absorption will be there, fast atom bombardment, and final will be MALDI, that is matrix assisted laser desorption ionization. Then third type will be evaporative ionization technique. So here thermospray, then electron spray ionization, atmospheric pressure chemical ionization, then atmospheric pressure photoionization. These are all the different types of ionization techniques. In any one of these ionization techniques, we will produce the sample vapors into a ion form. That is the first part in the mass spectrometry. So here one by one we'll see. So first will be gas phase ionization. This will be the oldest and common method of ionization. So here samples will not here in all the cases samples will be vaporized before going to the ionization chamber. So here in this electron ionization you can able to see electron slit will be there and electron beam will be subjected to this and heating environment is there. So because of this high energy electron beams, all the vapors coming inside it will be converted into a ions. That is the thing mainly happening in this electron ionization technique. After then it will produce ions. Then it will go to the ion accelerating region where it will get accelerated into the same energy. Then these ions will be going into the stream where we are applying strong magnetic field. Then it will reaching the analyzer tube. Then you can able to detect the ions according to the mass to charge ratio. So mainly what is happening in electron ionization is the energy will be you can able to see 6700 kilojoules. The energy will be okay. So here you can able to because of this high energy radial cation ions that That will be because of this high electron beam is applied, no? So because of this high compound, so the vapors will be coming inside. So the because this vapors will be, so because of this high energy electrons, the whatever vapor is coming now, that will converted into a smaller fragments. Okay, this is technique is called fragmentation. So you can able to get molecular ions as well as the daughter ions. So because of this electron impact, no, so this will be high energy technique. Okay, so this will be a hard ionization technique. So here you can able to see the pressure handling will be 10 per minus 6 to 10 per minus 5 tar. This is the electron ionization technique. And next will be chemical ionization technique. So here, with the help of some chemicals, ions will be produced. So normally we can use alcohols, amines, ethers, esters, amino acids, and these are all the chemicals which will be used for ionization. So the carriers will be used here is methane, ammonia, isobutane. So it come under the category of soft ionization technique. So here you can able to get intense peak of molecular ion. So here, because of this, if for example, if you are using methane as a carrier gas, so you can able to get here. Here you can able to get primary ions as well as secondary ions. So mostly primary ions will be CH4 plus and CH3 plus. Secondary ions will be CH5 plus and C2H5 plus. So after the production of secondary ions, that will be reacting with the analyte and it will produce your sample ions. Okay. Then these sample ions will be reaching the, I mean, accelerated and it will reaching the detector after the application of a strong magnetic field. So in this diagram, you can able to see. So in this diagram, you can able to see the reagent gas that will be coming in the topmost sector. You can able to see analyte A will be inside and M will be your reagent gas. Mostly methane is there, no? That M plus will be M star and A will be analyte. So that will be the secondary ions produced with the help of reagent gas. That will be reacting with the analyte. Okay. Then because of this, so after production of this ions, this will be accelerated, and then application of magnetic field will be reaching the deflection chamber and reaching the detector. Okay, then we can analyze which compound it is. So what you can able to see how the reaction will be. So first CH4 and the ions will be there. You can able to get methane molecular ion. Next, methanium formation. Then this methanium formation, after that, this will be analyte reacting with your analyte. So then protonated analyte will be there. This protonated analyte, that is M plus H the whole plus. This protonated analyte only it will be accelerated and finally it will be go through the deflection chamber. Then finally reaching the detector. Okay, this is happening here. And the third will be ionization technique will be matrix associated laser desorption ionization, that will be MALDI. So here some of the samples no we cannot able to vaporize. So that type of samples which can be analyzed with the help of ionized with the help of these techniques. So here we'll be having some chemical matrix. Okay, so the sample which cannot able to convert it into a vapor form. So that will be mixed with this chemical matrix and it will be deposited on a plate and it will be dried, and after the crystallization this can be used for ionization. So here the whole compound, the mixed one will be the sample with the matrix, everything will be used for ionization in this technique. Okay, this is a soft ionization technique. So here what is the thing is happening here, so the in the plate everything will be coated, deposited, and dried and crystallized. Then here this will be in this plate, will be applying a laser. So because of this laser application on the chemical matrix where the sample will be inside. Okay. So then they ionizes and desorbs ions. Okay. Then this will be converted into a vapor state. Then it will be detected with the help of mass spectrometry. Here, what are all the components in this technique, MALDI? So, one is ionization of an analytes and sorting of ions, detection of ions. So, what is the matrix? What are the properties of the matrix that you should know? Okay, so it is a small organic molecule will be used as a matrix here, and it will facilitate ionization process by absorption of laser light or UV light. Okay, then the properties will be of low molecular weight. It should not react with your compound analyte and it will be acidic in nature. Then uh, where we are applying strong laser beam or UV light. Okay, so that should be incompatible with this, and it should be of polar functional group. So these are all the properties of the matrix. And example will be two different examples I have given: dihydroxybenzoic acid, and next will be that is DHB. Second will be CHCA, that is cyanohydroxychinamic acid. The structure will be there. You can able to see. So these are all the chemical compounds. You can call it as a chemical matrix. So to this matrix, we'll be mixing our sample and we'll be coating in the plate. That will be used for ionization. Okay. So next we'll be going to discuss about one specific technique in the mass spectrometry, that is MALDI-TOF. So here you can able to, the ionization technique will be MALDI and the deflection analyzer will be time of flight. So that is the thing you'll be using here. So you can easily find out these two things specifically. This is one type of mass spectrometry. Okay, so here the main application of this MALDI-TOF will be specifically if you want to find out the molecular weight of a peptide, proteins, and antibodies. And the protein molecules also, that size will be up to 300 kilodalton. So if you want to analyze these different specific compounds, you can use this MALDI-TOF spectrometry. It is only used to find out or analyze the organic molecules. So here the ionization technique will be MALDI type of ionization technique. So it involves laser striking a matrix of a small organic molecule to make a large analyte molecule. Okay, into a gaseous ions molecule that will be converted into a gaseous ions without any fragmentation. That will be the major uniqueness in the technique. And TOF is the time of flight. So this is the mass spectrometric method that separates ions according to the mass to charge ratio, and it determines mass to charge ratio by the time where it reaches the ions, because according to the mass to charge ratio it will reach the detectors of different time intervals. So that is why time of flight the name will be there. It is the type of analyzer. So in this technique, so here you can able to see how we can prepare the sample for this technique. So first the sample will be dissolved in some solvent at different concentration, that will be solution A. Then second will be the matrix separately. Okay, so our sample is dissolved in a solvent, that is solution A. Then second one is matrix. We have told no different types, DHA examples. So this matrix will be dissolved in a solvent, that is a solution B. Then at different ratios, the solution A and solution B will be mixed. So here some examples they have given 1 is to 1000. So according to these ratios, we have to mix these solutions and will be in a plate will be there. So there we have to coat it and it will be co-crystallized. So this is the sample precipitation in MALDI-TOF. Okay. Then here you can able to see how the ablation and ionization will be there. So the matrix is there, no matrix and light will be the target plate. Okay. So here you can able to differentiate which is our compound. So different ions will be produced and you can able to detect it. And the mass spectrophotometer, that is mass TOF, mass spectrophotometer. So it will accelerates ions in the electric field. So the electric field we are applying will be directly like it's a constant kinetic energy. Okay, so you know kinetic energy how to calculate half mv squared. Okay, mass and the velocity of the atom or a compound it will be reaching. And The advantage of the small d of technique will be, so you can use non-volatile molecules for ionizing low non-volatile compounds. You can use this matrix ionization technique. It is a soft ionization technique. You can use mixed samples also and sensitive samples also. And it allows the storage of solid samples, because will be crystallizing the in this technique we'll be crystallizing the samples. So storage of solid samples also you can prepare. And the next technique will be electron spray ion. This is the last technique, electron spray ionization. This is the here you can able to see different small droplets of your molecule or a solvent, like an aerosol particle. If you see, no, so like aerosol form, your sample will be injected here. Okay, so here the solvent contains a proton source which will protonates the basic size of the small molecule. So here as the because of this electron spray ionization, this aerosol spray, your sample will be produced as ions, and these ions will be accelerated, and it will be detected. So here it will be also soft ionization techniques. So here you can use high molecular weight compounds. Okay. Then here, narrow metal capillary held at high voltage, it will be there. So, because of this high ESI source, so we can produce ions. So, then here the solution containing analyte is first sprayed inside. So, after spraying, you can able to get aerosols, your sample ions in the form of aerosols. Then it will be converted, this liquid streams will be converted into a tiny droplets, and it will produce ions. So next, in the ion formation, so the reduction reaction will be happening here, because so small capillary will be there, correct? In the capillary tip only, your samples in the droplet form will be getting inside. Okay, wow. So it can able to produce multiple charged ions. So that is the uniqueness in this technique. So here the ions will be converted into a gas phase. So one is ion evaporation mechanism, and second one is charge residue molecule, CRM mechanism. You can able to see in these two mechanisms, you can able to produce ions. Single, simple positive charged ions. That is as desolvated ions. Okay. So solvent will be also there, and your sample will be also there. So now you, after this final end, no, you can able to get your sample ions. That is a desolvated ions, because after removing solvent particles, only your compounds will be there. Then it will be accelerated and it will be analyzed. So this is the simple examples. ESA, how it will be used and what is the application of ESA, how you can do. So here in this first diagram, no, for vitamin B detection. So it will be here the solvent will be methanol water. Okay. So along with vitamin B is there, no. So you can able to detect vitamin B. This is the ESA MS of vitamin B. Then second will be drug compound, cyclosporine A. So this can be detected with the help of mass spectrometry. Here the specific ionization will be electron spray ionization. Okay, this will be the application of this specific type of ionization. And you can able to see, this is the overview of this ionization. So electron spray ionization, you can able to see ions will be there, so slit will be there, and after that ions will be accelerated, and the quadrupole rod type of detectors will be there. Here you can able to see, that is a, we have mentioned, analyze the different types of analyzer. Time of flight we have seen. The next type will be quadrupole analyzer, so that will be there here. This is one more technique. So I think we have covered mass spectrometry in detail. So if you have any doubt, you can ask me. So next class we'll be going to discuss about X-ray and other type of structural elucidation techniques. Okay, ma, thank you.